Welcome to this lecture on allosteric enzymes and their kinetics. Today, we're going to explore how our cells tightly regulate complex biological processes. We will learn how certain enzymes act as biological switches, turning metabolic pathways on or off exactly when needed, ensuring our bodies maintain perfect balance. Let us dive into the microscopic world of cellular control. To understand allosteric regulation, imagine a bustling car manufacturing factory. Workers assemble parts on a main conveyor belt, which represents the enzyme's active site. Now, imagine a separate control room located far away. A manager there can speed up or shut down the assembly line without ever touching a car part. In our cells, allosteric enzymes work exactly like this managed factory. They have a main workspace for the chemical reaction, but they also feature a separate remote control panel called the allosteric site. This incredible design allows our bodies to tightly regulate biological processes rather than letting them run out of control. Now that we understand the basic factory analogy, let us look at how this control panel actually changes the enzyme. When a specific molecule, called a modulator, binds to the allosteric site, it forces the entire protein structure to shift between two distinct physical states. The first is the tense state, which has a very low affinity for the substrate. The second is the relaxed state, which has a high affinity and readily binds the substrate. Positive modulators push the enzyme into the active relaxed state, turning the factory on. Conversely, negative modulators lock it into the inactive tense state, shutting the factory down. Building on these structural shifts, there is another fascinating feature of allosteric enzymes we must discuss. These enzymes typically consist of multiple polypeptide chains, or subunits, working together. Because they have multiple subunits, they exhibit a phenomenon called cooperativity. This means that the binding of one substrate molecule to one subunit physically changes the shape of the neighboring subunits, making it much easier for subsequent substrate molecules to bind. It is a cascading effect. Once the first molecule attaches, the enzyme rapidly opens up, accelerating the reaction. This cooperative behavior is what gives allosteric enzymes their unique and powerful regulatory capabilities. Understanding this cooperative behavior leads us to see why these enzymes are absolutely crucial for our survival. They act as the primary pacemakers and regulatory checkpoints in our cellular metabolism. A classic real-world example of this is feedback inhibition. Imagine a long metabolic pathway where the final product acts as a negative allosteric modulator for the very first enzyme in that pathway. For instance, when your body produces enough of the amino acid isoleucine, those isoleucine molecules physically bind to the allosteric site of the enzyme threonine deaminase. This shuts down the production line, preventing the waste of precious energy and maintaining perfect biological homeostasis. This elegant biological control mechanism directly affects how we measure the enzyme's speed. Because allosteric enzymes have multiple interacting subunits, they do not follow the classic, simple Michaelis-Menten kinetics that produce a standard hyperbolic curve. Instead, when you plot their reaction velocity against substrate concentration, you get a distinct S-shaped curve known as a sigmoidal curve. This sigmoidal shape is the mathematical signature of cooperativity. It visually demonstrates that the enzyme acts like a switch, remaining relatively inactive at low concentrations, but turning on very rapidly once a certain threshold of substrate is reached. To mathematically describe this S-shaped cooperative behavior, biochemists use a modified version of the Hill equation. Let us break down what each symbol means. V0 represents the initial velocity of the enzyme-catalyzed reaction. Vmax stands for the maximum possible velocity, achieved when every single active site is completely saturated. The variable S in brackets represents the concentration of the substrate. K0.5 is the substrate concentration at which the reaction velocity is exactly half of Vmax. Finally, the exponent n is the Hill coefficient, which measures the degree of cooperativity. Physically, if the substrate concentration increases, the velocity increases. If the K value is lower, the enzyme reaches maximum speed faster. And if the Hill coefficient increases, the switch-like behavior becomes even more abrupt. Now that we have our formula, let us walk through a practical calculation to see how cooperativity affects an enzyme's speed. We are given a maximum velocity of 120, a K0.5 of 4, a Hill coefficient of 3, and a substrate concentration of 2. We want to find the initial velocity. First, we substitute these values into the modified Hill equation. Because the Hill coefficient is an exponent, we must raise both the substrate concentration and the K value to the power of 3. 2 cubed is 8, and 4 cubed is 64. Multiplying the numerator gives 960, and adding the denominator gives 72. Dividing these gives a final initial velocity of 13.33 micromoles per minute. Notice that at half the K value, the velocity is far less than half the maximum velocity. This physically means the enzyme is waiting for a stronger signal before fully activating. Let us look at a second example to truly appreciate how sensitive these enzymes are to small changes. We will use the exact same enzyme, but this time the cell has produced a bit more substrate, raising the concentration from 2 to 6 millimolar. We substitute the new substrate concentration of 6 into our formula, keeping all other constants the same. We calculate the cube of 6, which is 216. Multiplying 120 by 216 gives a large numerator of 25,920. Adding 64 and 216 gives a denominator of 280. Dividing these yields a new initial velocity of 92.57 micromoles per minute. This shows a massive jump in reaction speed. Physically, this means the enzyme has fully activated, acting just like a biological switch that has been flipped on. By comparing the results from our two examples, we can mathematically prove why allosteric enzymes are considered biological on and off switches. Let us set up the comparison. In our first state, a substrate concentration of 2 gave a velocity of 13.33. In our new state, a substrate concentration of 6 gave a velocity of 92.57. If we look at the ratio of the substrate, we see that the concentration merely tripled, increasing by a factor of 3. However, when we calculate the ratio of the velocities, we divide 92.57 by 13.33, which gives us approximately 6.94. This mathematical reasoning shows a highly disproportionate response to the substrate. Let us finalize this proof on the next slide to see what this means for the enzyme. Continuing our proof from the previous slide, we arrive at a striking final result. A mere 3-fold increase in substrate concentration caused an almost 7-fold increase in the enzyme's velocity. This massive, nonlinear jump mathematically proves the power of positive cooperativity driven by a Hill coefficient greater than 1. It demonstrates that allosteric enzymes act like highly sensitive triggers. They remain mostly inactive at low concentrations, conserving resources, but turn on with explosive speed once a specific biological threshold is crossed. This perfectly illustrates the switch-like sigmoidal nature of these enzymes, proving exactly why our bodies rely on them for precise metabolic control.
Let us visualize everything we have learned by looking at this kinetic graph. The horizontal axis represents substrate concentration, and the vertical axis represents initial velocity. The solid black S-shaped curve in the middle shows the normal allosteric enzyme. Notice how it starts slow and then accelerates. Now, look at the dashed blue curve to the left. This represents the enzyme with a positive allosteric modulator, or activator. It shifts the curve left, meaning the enzyme reaches maximum speed at a much lower substrate concentration. Conversely, the dashed red curve on the right shows a negative modulator, or inhibitor, which shifts the curve right, requiring much more substrate to activate the enzyme. This diagram beautifully summarizes how modulators fine-tune enzyme activity. We have covered a lot of ground today, so let us summarize the key takeaways. First, allosteric enzymes are the sophisticated control valves of cellular metabolism, regulated through separate allosteric binding sites. Second, their activity is controlled by shifting between an inactive, tense state and an active, relaxed state. Third, their multiple subunits allow for cooperativity, which produces their signature sigmoidal, S-shaped kinetic curve, rather than a simple hyperbolic one. Finally, we saw how the modified Hill equation perfectly models this explosive switch-like behavior. You now have a solid understanding of how biological systems maintain strict, efficient control over complex metabolic pathways. Great job.